Hi, uh, hello everyone. This is Vasuki from Shanto Biology classes. So in the last class, we have discussed about an overview of the CUET PG 2025 exam. And in today's class, we are going to discuss about the CUET PG 2025, particularly the subject of zoology. In this session, I'm going to focus on the syllabus analysis of the zoology. Okay, so without any delay, we'll enter into the session. uh, these are all the same thing which we have discussed in the previous classes. So once again, let me brush it up. So the mode of this particular exam will be on online, and the medium of this exam will be conducted in the English and Hindi language. Both of these language will be included in this exam, and the duration of this exam will be 2 hours, and the exam will be conducted annually once in a year. And the question type of questions will be the multiple choice question. We have an options in these questions, and total number of question will be 100 question. And as we discussed in the earlier classes, one question carries 4 marks. One correct answer carries 4 marks, and total marks will be 400. Okay, and we have the negative marking. As I said, one for each wrong answer, 1 mark will be deducted from the marks which we have scored. Okay, and there will be 2 sections in this particular exam, and in section A will be this is somewhat related to the language comprehension, verbal ability, and general knowledge, mathematical ability. This somewhat related to the analytical skills, okay, of our mind. And section B is somewhat related to our subject which you are getting chosen. Okay, and regarding the subject of zoology, we have totally 14 units. Okay, we have 14 units, and unit 1 is related to cell biology. We know that cell biology is completely related to the study of cell, and this also includes cell organelles and their functions, how each organelle is going to do their function. And it is also related to cell signaling and cell communication. So there will be a lot of minor topics coming under this particular cell biology, and the most important units also. Okay, and the unit 2 will be the biochemistry. Okay, and this biochemistry, it is somewhat related to biomolecules, which is carbohydrates, proteins, lipids, vitamins and minerals. And we are also in biochemistry, we have a lot of cycles in that. Okay, and particularly, for example, in carbohydrate metabolism, we have a glycolysis, glycogenesis, glycogenolysis, and gluconeogenesis. So all these cycles plays an important role when it comes to the biochemistry. So these are all the important area. And in molecular biology, we are going to read the central dogma, which is replication, transcription, translation. And more specifically, we are also going to read a post-translational modification and post-transcriptional modification. So the entire thing will be completely covered in the molecular biology, also the most important unit. And in unit 4 and 5 is somewhat related to the ecology and evolution. So when it comes to the ecology and evolution, if we know the concept, we can answer any type of questions. See, biochemistry, molecular biology, cell biology, these are all the crucial area. Uh, if we know the accurate answer, we can choose that particular question. But in case of this ecology and evolution, even if we know the smaller amount of background information, we can easily answer the question. Okay, so if we read this ecology and evolution thoroughly, definitely these are all the questions we should not leave. Okay, so ecology and evolution is somewhat related to that. And unit 6 is related to the biodiversity and environment conservation. So when it comes to the biodiversity and environment conservation, we have to read the area. We have to read tropical forest, tropical rainforest, temperate forest, and what are all the hot spots. And we have to read about the national parks and wildlife sanctuaries. So what each sanctuary is actually, what is actually they are conserving, okay, which particular animal. And we also have to focus on the IUCN status, okay, whether that particular organism will be extinct or critically endangered. So these are all the area where we can expect the questions. And what are all the types of conservation we are having, like in situ conservation and ex situ conservation, and what are all the types which we are following in situ conservation, what are all the types we are following in ex situ conservation. So these are all the things we are going to read in this particular unit. And unit 7 will be the immunology. Okay, immunology, once again, the most important area. Okay, when it comes to the immunology, we have to read how our immune system is going to work, and how each cell, what are all the cells is involving in this particular defense mechanism, and what are all the types of defense we are having, what is innate immunity and what is an adaptive immunity, and what are all the types which is placed under the innate immunity, and what exactly is adaptive immunity. And after that, what are all the lymphoid organs, what is mean by lymphocytes. So these are all the things we are going to discuss in the immunology. Okay, and in unit 8, genetics, the most important area is genetics. In this area, we have to start from the Mendel concepts. Okay, as we know that father of genetics will be Gregor John Mendel, and we are going to read what are all the experiments done by the Gregor John Mendel and where it is going to end. Okay, regarding the monohybrid cross, dihybrid cross, and trihybrid cross. After that, we are going to read about the linkage and crossing over, gene mapping, recombination, so recombination frequency. So all these will be covered under the genetics, the most important, where we can find many questions. And biotechnics. Techniques is somewhat related to the instruments we are using. Okay, whenever we are doing any research, instruments plays a crucial role. And when it comes to the techniques, we have to know the name of the technique and their working principle and what is their applications. If we read all these things, that's all about the biotechnics, that's all about the technique. Okay, and if you find any graphs, we should not leave that graph as usual. And invertebrates and vertebrates, so the entire living world can be categorized into two types, like vertebrates and invertebrates. In invertebrates, what are all the phylum we are having? So when we can classify like that, taxonomical hierarchy, we have a domain and we have a kingdom, phylum, class, order, family, genus, and species. So we can categorize a classification. We have start this particular topic from the classification and end with the class, particularly the genus and species. See, when it comes to the core data and non-core data, you have to know what are all the phylums, and in each phylum, what will be their unique characteristic features. So if we read like this, like classification, for example, take for an example, phylum Porifera. In this phylum Porifera, we should know the general characteristic features. After studying about the general characteristic features of Porifera, we have to concentrate on the classification of the Porifera. We know that Porifera will be the phylum which is coming under the kingdom Animalia. And after that, this Porifera will be categorized into different class. And we also know that what are all the class will be classified under the phylum Porifera. And also we should focus on the examples. When it comes to the non-core data and core data, we have to thorough with their examples. Okay, and after finishing that, what will be their unique characteristic features? So with respect to the Sporifera, we have an important characteristic features, and we can't find that characteristic features in any other kingdom, like canal system. And this is the characteristic phenomena which is present only in the phylum Porifera. Like in the same way, we have the unique characteristic features regarding all the phylum. For example, water vascular system, which is present only in the phylum Echinodermata, right? Fine. So if we read all these things, the entire invertebrates and vertebrates will be very easier. And developmental biology. Developmental biology, we have to start from the gametogenesis. Okay. And what is spermatogenesis? What is oogenesis? And after that, we have to study about fertilization. Now, as a result of fertilization, we are going to get a zygote. And after that, it will be enter into the cleavage. And as a result of cleavage, we are going to get a stage of blastula and gastrula. And what is blastulation? What is gastrulation? And after that, how an individual organism, how one group of cells is going to give rise to an organ, that is organogenesis. So this is completely related to the developmental biology. biology. Also, we have to focus on some of the genes. Which genes is responsible for the development of this? Which genes are under the control of this particular function? Okay, like that, we have to focus on the developmental biology. Biostat, it is somewhat lesser important area, because if we read about a selected area, we can easily solve these questions like mean, median, mode. If this three are the most basic topics. So whenever you're going to start reading about this biostat, you have to focus first thing, that will be the mean, median, and mode. Okay, and after finishing that, you have to focus on so in these things you have to focus the formula, and after that you have to read about the standard deviation, and you have to these are all the topics which you have to focus first when you start reading the biostat, because these are all the lesser important unit. Okay, and animal physiology. Okay, animal physiology, it is a most important unit as usual. You have to start reading the system. Okay, like circulatory system, reproductive system, and hormone, endocrine system, and excretory system, nervous system, chemical coordination. There are we can scrutinize this 9 important topics under the animal physiology, like systems, like all the systems which I have mentioned before. Okay, if you finish all these things, the entire animal physiology will be over. And recombinant DNA technology. This is an important technology, and in this technology, we have to read about the vector. Okay, what is vector? What are the types of vector? And we have a different types of vector, and in each vector, we are following a different mechanisms, and we have to read about the enzymes like restriction endonuclease, DNA ligase. So these are all the enzymes and vectors plays an important role here. And after that, what is foreign DNA? What is donor DNA? What is the recipient DNA? And what, as a result of this technique, what we are supposed to get. Okay, and what are all the applications of this particular recombinant DNA technology? So all these things will be covered under the recombinant DNA technology. So these are all the list of 14 units, and also we are going to focus the most important units first, and after that we are going to see the lesser important units. So I have given 7 important units which we have to focus as the first priority. So the first unit will be the cell biology. As I said before, we have to focus overview of cells. What is a cell theory and who proposed the cell theory? Starting from that, and we have to enter into the cell organelle. Okay, and initially, without reading the plasma membrane, we should not enter into the other topic. So after reading the overview of cells, you should know all the entire information regarding the plasma membrane. Okay, because this plasma membrane is everything. Okay, entering once we have, if we want to enter into the cell, and if you want to eliminate from the cell, everything will be crossed through this plasma membrane. So we have to read this plasma membrane first, and after that we have to enter into the cell organelles. We have a different cell organelles like endoplasmic reticulum, Golgi complex, ribosome, lysosome, centrosome. So these are all the important cell organelles we are having, and you should know that what are all the things they are following and how they are communicating, and from the nucleus through which organelles everything is getting sorted. So everything will be dealt in the cell organelles. And the nucleus, this will be the central part of the cell. And the most important thing will be it consists of DNA, a genetic material. So we know that. And when it comes to the nucleus, you should focus on the picture, like how nucleus is supposed to be. It will be covered as covered by an inner membrane and outer membrane, and it composed of a what is nucleoplasm, and what is nucleolus, and what is nuclear pore, and what is nuclear envelope. So all these terms is also an important thing, and how and through which area all the materials which is present in the nucleus will be exit from the nucleus and entry into the nucleus. So all these things will be discussed in the nucleus. And cell division. And the most important thing, cell division. Cell division, we have a mitosis and meiosis, and cell cycle. See, there will be a difference between cell cycle and cell division. Cell cycle is somewhat related to a phase which is coming under the cell cycle. Cell division means mitosis, and other phase will be a G1 phase, G0, S phase, interphase. All these will be collectively called as a cell cycle. And in that cycle, we have an M phase, and the M phase is nothing but a mitotic phase. In mitotic phase, we have a different phase like prophase, anaphase, metaphase, anaphase, and telophase. In these things, we are going to, the cell is getting divided, okay, and all the preparatory phase will be nothing but this G1 phase and S phase. So cell cycle and cell division, the most important thing we should not left at any cost. And cell signaling, it is somehow one cell is getting information from the other cell, how the cell is getting communicated, and through what are all the junctions we are having, what is tight junction, what is desmosome, what is hemidesmosome. So all these things will be discussed under the cell signaling. Okay, so these are all the important things which we have to be covered under the unit 1 cell biology. And the second unit will be the biochemistry. If we study this carbohydrates, lipids, proteins, nucleic acids, and the entire biochemistry will be getting covered. Okay, in this carbohydrates, we have to focus the carbohydrate chemistry and carbohydrate metabolism. Like in the same way, lipids, proteins, we have to cover their metabolism and chemistry. Okay, for example, in carbohydrate chemistry, we are supposed to read about the what is monosaccharide, what is oligosaccharide, what is polysaccharide, and how these sugars are getting how polysaccharide is getting hydrolyzed into monosaccharide. So all the structure will be covered under the carbohydrate chemistry. But in metabolism, we are supposed to read about the cycles like glycolysis, as I said before, glycolysis, glycogenolysis, gluconeogenesis. Okay, all these things will be covered under the metabolism, how they are metabolized and how they are taken to the cell. Okay, how they are getting converted into the energy. All these things will be covered under the carbohydrate metabolism. Like in the same way, the proteins, lipids, and nucleic acids will also getting covered here. Okay. Now, when it comes to the enzymes, we have to read about what is enzyme, what are all the types of enzyme, and what is enzyme kinetics. Enzyme kinetics plays a crucial role. And when it comes to the enzymes, we are also supposed to read about the enzyme inhibition. What is competitive inhibition? What is non-competitive inhibition? And we are also having a lot of graphs in this particular enzyme topic. So we should not leave these graphs. And if we read about all these things, the entire biochemistry is over. That's all. And after that, in unit 3, the most important unit, the molecular biology. And in this unit, we are going to read about the DNA replication and nucleic acids. We have to start about the start from the nucleic acids. And we have two different types of nucleic acids. One is RNA and DNA. That is a ribonucleic acid and deoxyribonuclease. And what is the difference between this ribo and what is the difference between this deoxyribo? And starting from their structure, we are going to read about how they are made up of. These nucleic acids is somewhat polynucleotides. And what is nucleotide? Nucleotide is somewhat, it is made up of sugar, base, and phosphate, and these three things will be combinedly called as a nucleotide. And what is the difference between nucleotide and nucleoside? So these are all the basic things we are going to cover under this nucleic acids. And after that, when we come to know that the DNA is a major genetic material, we humans is having as the genetic material, we are going to start from the DNA replication, transcription, and translation. Okay, these are all the central dogma of molecular biology. And what is post-transcriptional modification? Post-transcriptional modification, whether they are, and when it comes to, when we are reading about this replication, transcription, and translation, we are also going to focus where it is getting actually takes place. Replication takes place in nucleus, transcription in nucleus, and how they are varying from the prokaryotes and in eukaryotes. In prokaryotes, everything will be done in the cytoplasm, but in the in eukaryotes, everything will be done in the nucleus. So when it comes to the place, how it will be very, we have to compare them from the prokaryotes to the eukaryotes, and what are all the transcription factors, translation factors involved in the transcription and translation. And also we have to read for that for both prokaryotes and eukaryotes. Okay, so these are all the things will be covered under the replication, transcription, and translation. And we should not stop up to that. They're also having a we are also having that before the translation is going to occur, there are also some of the changes, modifications will be occur after the replication, sorry, after the transcription, and that will be called as the post transcription modification. And after the translation, post translational modification is also when it takes place, and what actually is taking place there. So everything will be dealt there. And gene regulation, and after finishing that, how the gene is getting regulated, everything will be, if we read all these topics, the entire molecular biology will be covered. And in unit 4, ecology, as I said before, ecology is the most easiest area and also the most scoring area. Okay, when we should not leave this ecology. And we have to read about the ecology, population, community, ecosystem, and applied ecology. We have to start from the individual, and we have to individual into population, community, ecosystem, biosphere. Okay, if we covered all these things, the entire ecology will be covered. Okay, and in unit 5, vertebrates and invertebrates, this will be there will be a lot of things will be covered under the, has to be covered in these vertebrates and invertebrates. And when we focus, so as I said before, when it comes to this vertebrates and invertebrates, porifera, what are all the phylums, whatever the phylums we are having, we have to focus on their general characteristic features. This is the first thing we have to focus whenever we are start reading about this vertebrates and invertebrates. And after finishing the general characteristic features, we have to focus on their classification. Okay, and after finishing the classification, each phylums will be having their own unique characteristic features, and if we read that, the entire phylum will be covered. And if we read all the phylums, the entire vertebrates and invertebrates will be covered. Okay, and that's all about the unit 5. And 6th, the priority unit will be the animal physiology. When it comes to the animal physiology, if we covered the important system, that's all about the animal physiology. We should know what are all the tissues, and what when it comes to the tissues, we should know what are all the types of tissue, like epithelial tissue, connective tissue, like that. We have 3 different types of tissue, and in epithelial tissue, we have different types of tissue like squamous epithelium, cuboidal epithelium, and columnar epithelium. And also, we should have to read about the examples where this exactly the squamous epithelial tissue will be present. Okay, if we read about the types of tissue and subtypes of this tissue and examples, that's all about the tissues. And we have to know about the difference between bone and the cartilage. Okay, and after that, we have to come to the important systems like nervous system, muscle physiology, reproductive system, endocrine system. There are also some of the other important systems like excretory systems is there. So if we, circulatory system is there. If we cover all these important system, the entire animal physiology will be covered. Okay, next. Unit 7, that the 7th priority unit will be the genetics. Okay, that is Mendelian genetics, starting from the Mendel, like monohybrid cross, dihybrid cross, and trihybrid cross, and what are all the suggestions we have learned from these crosses, and how these crosses will be help in the applications of genetics, and what is linkage and crossing over, what is mutations, what are all the types of mutations, and we have to read about the sex determination, how the sex will be determined in case of human beings, how the sex will be determined in case of insects, and what is XX type, what is XO type, what is XY, what is ZZ, what is ZW. Everything will be dealt in the sex determination. And what is extra chromosomal inheritance, what is recombination. Okay, recombination, the most important things. If we cover these things, majority of the topic under the unit genetics will be covered. Okay, so these are all the most important 7 units we have to give as our first priority. We have to read as our first priority these 7 units. Okay, only after finishing the 7 units, we have to move on to the other units. Okay, so that's all about the syllabus analysis for zoology, and we'll meet up in the next session. Okay, thank you, everybody.